Hi, my name is Shivansh. As she said, I'm fifth year PhD student in her lab, hopefully my final year. And today I will talk about signatures of selection in the genome. So I have attended this school several times, but I have never presented it in an offline setting. So this is my first time. I'm nervous and excited. So please be kind. So what do I mean by signatures of selection? So let's say you have a population of some living organism, let's say birds, that usually lives in grasslands, and it has now entered into a new environment, let's say high altitude. Right. After some time, this population would adapt to this new habitat, and you are interested in understanding the genomic basis of this adaptation. What are the genes that confer higher fitness in this new habitat? What are the specific mutations that increase its fitness in the new habitat? So the traditional way to go about this is to first find out the phenotype on which selection is acting. So you'll take the population from the grassland, take the population from the high altitude, measure a bunch of traits, and try and identify which trait is under selection, right? Next step would be to try and find the genotype or the underlying genomic basis of that trait. But this is a long and tedious process. And since high throughput sequencing is now easily available and it's cheaply available, there is a direct way of looking at the genome of an individual to identify where selection is occurring, on what genes, on what mutations selection is occurring. And that's what I'm going to talk about today. The best signature of selection is selective sweeps, which has been introduced to you by both Kavita and Krishnamekh. I will talk about 3 distinct features of selective sweep that you can identify in the genome. One of those features is measured using Tajima-D, and this is what we'll do in today's tutorial. And if time permits, I will also talk about FST, which is a special case. It's a metric to measure divergence between populations. And in special cases, you can use this to get some information about selection in the genome. And again, if time permits, I will talk about experimental evolution studies where people use evolve and resequence techniques to identify what regions of the genome are under selection. So what is a selective sweep? Can somebody quickly explain or define what a sweep is? I hope everybody knows what a selective sweep is. People have talked about this in the school several times now. In just the allele frequency, or is there anything missing in that? So yes, there will be an increase in allele frequency at the selected site, but selective sweep is determined by the change in allele frequencies around that selected site because of linkage. You would have, this is a driver mutation. So let's say this is a population, and all these gray bars are genomes, and all these blue blocks are polymorphisms. And in some new environment, this red polymorphism is under selection. Now, after selection, the frequency of this red polymorphism will increase in population. But along with that, anything which is linked to this polymorphism, that will also increase in the population. This process of hitchhiking is called selective sweep. Since usually the genomes are extremely large, you need to identify, in those millions of base pairs, you have to identify where selective sweep is occurring. And so I will talk about 3 distinct features of this selective sweep that you can use to identify where selection is occurring. One of the most intuitive features is decrease in genetic diversity around the selected site. This is extremely intuitive. So again, this is a similar graphic as previous one. All of these horizontal bars are genomes. All of these blocks are nucleotides, and polymorphisms are in yellow, and the selected site is in red. Now, before selection, you would have all kinds of polymorphisms existing in this genome, right? After selection, this red will increase in frequency, and along with that, everything that is linked to this red, which is these 5 yellow polymorphisms that you see, those will also increase. and everything else will be lost. And so you see, overall, there is a decrease in diversity between these two features, between these two events. After selection, the overall genetic diversity has decreased. You can measure this if you scan the whole genome. You can identify what are these regions where you have very low genetic diversity than what you would expect by chance, and that is what we'll do using Tarjuma's D in today's tutorial. Can you think of any other feature just by looking at this left panel? Can you think of any other feature which you can use to identify a sweep in the genome? I'll give you a hint, linkage. Yes, yes. So you would find that SNPs that are within the sweep are more likely to occur together, whereas a SNP which is in the sweep compared to something which is outside, those two are not likely to occur together. And so this high linkage will increase linkage disequilibrium within the sweep, and that's another distinct feature of selective sweep. Can you think of any other feature? This third one is not very intuitive, so I don't really expect you to know, but you can try. It's actually given here. Yes, what about mutations? No, selection is not removed here. It's just that some time has passed since selection. And so new mutations that do not really affect the phenotype are able to come up and survive in the population. So this will change the site frequency spectrum. Shweta was talking about this in yesterday's session. Does anybody know, or does anybody not know what SFS is? Site frequency spectrum? Sorry, what? Okay, so site frequency spectrum is basically allele frequency distribution. You have here allele frequencies on the x-axis. Forget the numbers, it ranges from 0 to 1. The paper that I took this from has allelic classes, so just like ignore. Here are the rare alleles, here are the intermediate alleles, and here are the common alleles. What do you expect in a population that is not undergoing selection? You would have a distribution like this, where you would have some rare alleles. You would have a large number of rare alleles, but you would have very few intermediate or common alleles, right? But after selection, after some time has passed, right after selection, you'd suddenly see a drop in the intermediate alleles. Why is that? That is because rare alleles will become more rare because diversity is lost, and common alleles will become more common because they are hitchhiking with a beneficial allele, right? So this change, this shift in site frequency spectrum is also a very good signal of selective sweep. Does anybody not understand any of these three features? Does anybody want me to go through these again?
But you said new mutation will appear in the population and which will increase the frequency of that new mutation, right? Which is advantageous to the population or beneficial.
The new mutations need not be beneficial. They will survive as long as they don't interfere with the phenotype which is adaptive. So in this example here, the red polymorphism is responsible for the adaptive trait, right? All these blue ones that have come up after selection are not changing the phenotype in any way, and so they will survive. They will basically break that haplotype, and so you will see erosion of this sweep signal. And this erosion will change the site frequency spectrum in a very specific way. You will see an increase in rare alleles because all the common alleles are lost, and only those that have come up recently will survive. And so here in the gray, you see a neutral expectation. But after a sweep, you suddenly see all these rare alleles going up, and all the common alleles also going up. These are the ones that are hitchhiking, right? Is this clear? Okay. So there are different tools to identify all of these three different features. I will talk about Tajima's D. It's a metric which is used to measure genetic diversity. So I will, yes.
So these SFS that you're showing, these are the SFS in very large times after sweep has finished and
Yes.
so on.
Yes.
Would you know of examples where people calculate these things and the sweep is ongoing?
Yes, yes. So in experimental evolution systems, there are lots of studies that have looked at the rate at which a sweep signal degrades.
No, the SFS, the non-equilibrium SFS, that is, sweep is still going and you measure SFS at that time. So I think the one that you're showing is after the sweep is finished.
I am showing here both. So I'm showing here the neutral expectation, and I'm showing here what happens right after a sweep, and I'm also showing here what happens specifically in a very strong positive selection or a very strong negative or background selection.
The sweep is ongoing. It's not finished.
It's not finished.
Okay, so while the sweep is ongoing, at that time if I calculate SFS,
That will not work, right? Because you don't have enough new mutations that have come up. So this signal will only come up after some time. So here in this example, in this panel, you see if you look at the sweep here, if you look at the site frequency spectrum at this stage, you will only see a sudden drop in rare alleles and an increase in high like common alleles. But only after some time, you will see that the other side of this distribution is also going up. Just because
frequency is increasing, right?
sorry, what?
The frequency of the beneficial mutant is increasing.
Yes.
So let's say it takes 100 years for it to finish, but I want to sample at 50 years,
Yeah.
right? So it's still an ongoing sweep.
But is it enough for new mutations to come up?
So instead of 50, you can take 75 or something. So I'm
Yeah.
saying that not at the end of sweep, but at least theoretically, we do calculate non-equilibrium SFS. So whereas you're calculating equilibrium, when the sweep is finished.
Yes.
So what I was asking is, do you know of any data when the sweep is ongoing and it's not the same signal? I know for
From experimental evolution systems, I know that there are studies that have looked at sweeps that are ongoing, and they have looked at site frequency spectrum in that. The only difference between the equilibrium SFS and that early stage SFS is that you will only see the signal in common alleles when it's ongoing, and as time passes, you will also see it towards the other side of the distribution. So rare alleles will also come up. So the left-hand side of this distribution is a result of mutations. The right-hand side part of this distribution is because of all the standing genetic variation, which is hitchhiking with the beneficial allele. And so this is already present when the sweep is ongoing. But these are not present when the sweep is ongoing. This will come up only as time passes and new mutations are able to accumulate. And so SFS will be like very clearly different between an ongoing sweep and a later stage of the sweep.
So are these data sets from viruses, for example, or are these from, like I know we had done some work with HIV, for ongoing sweeps, and I'm wondering where these, is it expensive to get these SFS, and you sequence a lot to get these SFS. So in the ongoing case, I can't imagine there be too many studies which will
There are COVID studies that have looked at ongoing sweeps, and there, like, it's very clear that the left-hand side of this distribution will only come up, like, only show as a signal at the later stages. You will only be able to identify a sweep in early stages using SFS only from this right-hand side of the distribution. Any other question? No. Can I ask a few questions? You have to think of confounding factors, right? Selection is one reason why you would see a decrease in genetic diversity. Can you think of any other process which would lead to this signal? Sorry, what? You have to be louder, yaar. Come on. Inbreeding. To some extent, yes, but there are other ways to identify inbreeding, like runs of homozygosity. Any other? That's the correct answer. I'm not saying that's the wrong answer, but can you think of? Can you be more specific? Yes, drift. Exactly. So a bottleneck would create exactly this signal, right? A bottleneck would erode most of the genetic diversity, And only those organisms that survive in that bottleneck will increase in frequency. And all the genetic diversity which was lost because of dead individuals will be lost from the population, right? So a bottleneck would create this signal as well as selection. Can you think of any process other than selection that can do this? Sorry, what? So I can't hear you. How would founder effects change linkage disequilibrium? I'm talking about the second one. So the first one, it's correct. So bottleneck and then expansions would eventually lead to this kind of a signal. I'm talking about this linkage disequilibrium. Can you think of processes other than selection that can increase or decrease LD between two SNPs other than selection? Yes. So what exactly is, so what exactly in the genome would lead to that signal? Do you know of any specific regions in the genome that have high or low recombination rates? Repeat regions, yes. Anything else? Centromeres, telomeres, they will have lower recombination rate than the rest of the genome, and so you will have high LD there. Any other process?
Translocation can also change.
Exactly, yes. So structural variants like inversions, translocations that inhibit recombination in a genomic region would also lead to this signal. And it's very important to know all of these confounding factors because ultimately when you find a signal, you should be able to attribute it to just selection, right? Can you think of any other process for the third feature? This is a bit tricky. Yes, yes. I was expecting somebody to, so not just change in the mutation rate overall, but even variability within the genome. So there are some regions that will have higher mutation rate. There are some regions that will have low mutation rate. And so SFS will be very specific to that region. And so sometimes while using this to analyze, to identify a selective sweep, you have to take into account this intragenomic variation in mutation rate. And there are tools that can do that for you now. I will not talk about this, but we can discuss this after this session if you want. Any questions? Okay, so I'll now move to Tajamaz-D. Tajamaz-D is basically going to, it's a metric which is going to look at genetic diversity in windows in specific number of base pairs across the genome. So you basically break down your genome into smaller pieces, let's say 1 kb or 10 kb, and then you ask what is the observed genetic diversity there and what is the expected genetic diversity there. And so I would like you to remember what Shweta was showing yesterday in those two genes where you had a specific number of expected SNPs and observed SNPs. And she was asking you which of the two genes do you think is under selection. And what she told you there was that if you have a high difference in expected and observed, such that observed is much lower than what is expected, there is a process which is actually eroding this, right? It's preventing new mutations to come up. And that's selection. And so we use Tajamaz-D, it's one of the metrics that we use to measure this in the genome. So how do we do this? For this, you need nucleotide diversity pi, which we then use to calculate Tajamaz-D. Pi is average pairwise differences among individuals. We'll use a dummy data to calculate this now. This is the formula. This is the observed diversity. This is the expected diversity. So what you're doing here is calculating so you're adding up all the average pairwise differences between individuals and then dividing it with the total number of pairs. I'll show you with a dummy example, that this would be more clear. Expected is calculated by looking at the total number of segregating sites in that window. So let's say this is a dummy data from Wikipedia. It's a 20 base pair DNA sequence of 5 individuals. And this is compared to a reference allele. Wherever you see a 0, the allele is same as the reference. Wherever you see a 1, the allele is different from the reference. So that's a single nucleotide polymorphism. Now what you have to do, you'll have to actually calculate this. What you have to do is calculate the pairwise differences between all of these. So between 1 and 2, how many differences are there? Yes, 1 and 3. Can you do this calculation? Can you like add all of these and divide it with the total number of pairs that are possible in these five individuals? Oh, exactly this? Oh, okay, okay. I'll just move ahead then. Right? Now we'll move to the second part of this equation. S is the total number of segregating sites. So you have 20 sites here, right? How many of these sites are polymorphic? Can you count? Like, I know you can count, but can you like tell me? How many? 4, exactly. So S is 4, and the denominator of this is basically just the summation of 1 over the total number of individuals, which is 2.08. So the second half, the expected diversity is 1.92. So D is 2 minus 1.92, which is 0.08. Now what does this tell you? What do you expect? If observed diversity is greater than expected diversity, what does that mean? That means D is positive. What does that mean? Yes, so there is more diversity than what you would expect by chance. Can you think of processes that can lead to that? Yes, with high migration rate can lead to that. Anything else? A very high mutation rate is not a very good answer because you are taking into account mutation rates in both of these, right? You are calculating the expected diversity based on a certain mutation rate, right? So mutation rate is probably not the answer. Anything else? Any specific kind of selection that you have heard of which can lead to increased diversity? Random mating is actually the opposite of the answer, right? Random mating will lead to this. Could push it to 0, not positive. Anything else? Directional selection is also not the answer because directional selection will decrease the observed diversity, right? So D will be negative, not positive. I'm talking about processes that will increase the observed diversity. Sorry, I can't hear. You'll have to. Disruptive selection. Yes, that's a good answer. Anything else? Does everybody know what a disruptive selection is? Does anybody not know what disruptive selection is? Like, you have to tell me, yeah, come on. Okay. Anything else? Any other form of selection? That will make D positive here. Sudden bottleneck will not increase observed diversity. So sudden bottleneck will actually make Tajamas D negative, not positive. I will come to that. I will also ask you what processes other than selection will lead to that. So yes, you've jumped the gun here. Balancing selection, exactly. So there are different types of selection, right? Frequency dependent selection, balancing selection, there is heterozygote advantage, there is disruptive selection. All of these will maintain more diversity than what you would expect by chance. And so D would be positive when you have selection of the sort which will increase diversity. Now, can you think of anything other than positive or negative selection which will make Taj-e-Masdi negative? That means observed diversity is lower than what you would expect by chance, other than positive and negative selection. Exactly. So we have discussed this in the previous slide. Bottleneck will lead to this. And so that's exactly what my next slide is. So let's say you have a population which is at some equilibrium population size, and suddenly there is the bottleneck. You'd see that at first, just when the bottleneck appears, you would have a slight increase in D, and then immediately after that, D becomes extremely negative. And so the signal of sweep that you're trying to identify would be same for a demographic event. And so if you want to publish a paper using this now, you have to do some demographic simulations and ask whether what you see, the negative Taj-e-Masdi that you see, is indeed because of selection, it's not because of demography. Right? So you have to control for demography when you are using Taj-e-Masdi. Is that clear?
That means if D is greater, it increases diversity, but if the population is small, then it leads to this kind of extinction or a sudden change in population will increase the diversity.
Yes, exactly. Nice, we are on time. Any questions? So now we'll use a dummy data to actually calculate Taj-e-Masdi, and I'll show you how you have to take into account lots of these things, not just the conceptual problems that we discussed, but also some technical problems that you have to look for when you are analyzing data of this sort, like high throughput, large data sets. So just as a summary, on the x-axis here are genomic positions. On the y-axis here are scale statistic of these three values. And you'll see that at the site of sweep, you have low variability, low Taj-e-Masdi, but high LD. And that's exactly what we discussed so far. Is this clear? I would also like to direct you to this review. It's great. It's a bit outdated now, but it would give you a very good primer for this kind of analysis. Cool. So now we'll move to the tutorial. Do people have VCF tools installed? How many of you have VCF tools? Nobody? Really?
Because there is some issue with that DCA. I tried to yesterday.
Do you have Windows? It doesn't work on Windows. You need an emulator for that. So nobody has VCF tools? Then we'll just skip through all the VCF tools steps and move directly to the RStudio steps. Cool? Okay. So what is there in the data that I've given you? I have sequenced 26 individuals from a population of this beetle. It's a diploid organism. Okay? I have taken out its DNA. I have sequenced it using paired end Illumina sequencing. And I have aligned those reads to a reference. After alignment, I have called variants. I've called all the SNPs that are there in all these 26 genomes. And the VCF file that I've provided you contains all of these variants. So VCF file is basically a large Excel sheet with these many columns. The first column is the chromosome position. First column is the chromosome number. The second column is the position on that chromosome. The third column is the ID. You will find this empty in my VCF because we don't have SNP IDs, but if you're working with humans, you will have a DB-SNP code. The reference allele and the alternate allele. So in this particular example, the reference allele has T and the new mutation is A. There's a quality score. There is a flag for filter where the filtration is I have a filtration step. I don't want bad quality SNPs, right? I only want to work with good quality SNPs. So that was exactly the VCF step that I wanted you all to do. If a SNP passes all my filtration criteria, you would see a pass here. info tag has a bunch of other things like what was the depth at which this variant was called, what was the quality of the read, lots of different things. There's a maximum likelihood estimation of whether you have called this variant correctly. So you'll find all of this information in this info tag. There's a format and there is like whatever. You will not be able to open this VCF file in your computer because these are large files. But if you use a Linux machine and if you use the head or tail function in the terminal, you will be able to see some part of this table just to see whether you have the correct format or not. Okay, cool. Since most of you can't do this VCS step, I'll just still go through. Sorry, I have to. I will still just quickly go through all the things that you can do with this very powerful tool. Basically, if you provide a raw VCF file, VCF tools can help you filter out bad alleles, things that you may have called as variants just because of sequencing errors. So you can have all kinds of flags here. For example, I have a minimum depth and a maximum depth flag. Minimum and maximum depth means, yeah. So minimum and maximum depth means the number of reads that correspond to that allele. If you have at least 5 reads that tell you that the SNP is true, you would consider that as a true SNP. If there are fewer reads than this, then you would not call it a SNP. Same with maximum depth, because there are when you sequence, you will sequence repetitive regions again and again, and so depth would be very high there, and so there is usually a max depth cutoff as well. You have quality scores, so that means that the read that actually goes and aligns to the reference has to have some quality threshold that it has to pass. If the quality is not good, you want to remove that read entirely from your analysis. And there are a bunch of other flags that you can use. I have added all the information about these flags in a VCF tools file, a text file, which is there in the data set. What else can you do? So this VCF file contains all the information about the SNP position and all the individuals, right? You can use this to do some population genetics. You can ask questions like, what is the frequency of each allele in this population? So you have 26 individuals, right? You can calculate allele frequencies. You can also calculate TS and TV summaries. So how many transitions do you have? How many transversions do you have? What is the ratio? And these are, as you heard in Deepa's talk on Wednesday, transition and transversions also inform you a lot about the kind of selection or the kind of processes that are affecting this population. Okay? You can calculate pi, the nucleotide diversity that we calculated in that Tajamaz-D slide. You have to remember that both pi and Tajamaz-D are windowed statistics. You can't calculate it for one site. You have to have a piece of DNA. Here, I have done it for 1 kb because it's a 165 MB genome. So if you want to do it for anything lower than that, it would take forever. Even at 1000, it takes up to 40 minutes to measure it for just one chromosome. I have given you just one chromosome file. But you can imagine if you want to do it for the whole genome, it takes hours, sometimes days. I have things that run for days. You can also calculate, what else can you do? That's it. There are a bunch of other things, but you can look around and explore the tool yourself. So now if you can open RStudio and upload the data sets that I've provided. Sorry, I have to. Can you see this? you'll have to change the working directory to the folder where you've installed the data, where you have downloaded the data sets. Are you able to do that? Just like raise your hands if you're not able to do that. I'll come and help you. Huh, so wait, have you downloaded this in your?
No, I will do it right now because.
Oh, it will not happen. This is like a 350 MB file. So maybe just try now. I might have a pen drive you can use.
Directly to the file, to the path which has our data file.
Yes.
And is there R script for the
So the R script is called lgx.r. I did not do it the way Shweta did it. I don't have a R markdown file. I use direct scripts like this. Were other people able to access data sets?
Open another window and that's working.
Because the file permissions are anybody with the link can view and download. So everybody should have access to it. Yes. 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 Just download the whole. No, just download the whole folder. SS tutorial. Please download the whole folder because here the files are like labeled such.
The data files folder.
SS_tutorial. Download the whole folder. Zoom. Control++. Okay. Oh my God.
took a while, that's all. Okay, there, there. Was all right. Why don't you just mirror your thing?
I don't do that because I don't want to show the desktop. It's kind of messy and weird. So
I think anybody can.
I'm just shy.
Okay.
Can you see this now?
So that first command there is set working directory.
So you change this path to the path of the folder, SS tutorial. No, just put the outside folder here. Yes.
Like, if you can demonstrate the initialization step that like we don't have the BCF tool now, but we can see that how it's going.
So if I start on this now, it will take around 30-40 minutes. What I wanted to show was all the things that VCF tools can do for you. So you provide raw VCF file that I've given. You can try it at home. If you have VCF tools installed, you can use those commands. I showed you those commands, right? You can copy paste those commands in a terminal and you will have outputs. You can view the outputs. You will see the filter tag will have pass or fail. Some of the snips will not pass this filter. Some of the snips will pass the filter. And so you can use this now corrected VCF for further analysis. So when you try and calculate allele frequencies and all, you will use the corrected VCF.
Like if I shot somewhere, there is any video or tutorial on the.
You will find lots on YouTube. Just like go ahead and like explore. Most of these things you have to explore on your own.
Yeah.
This is forget the guidelines. Sorry, I made that for the assembly. Have you downloaded this on your desktop? Go to RStudio. No, you need the whole folder. So maybe just download. Maybe download scripts as well. Yes. Open RStudio and open the lgx.r script. That is also. No. There are a bunch of files in that. Wait. Maybe you did not download it correctly. I'll show you. So this is that whole folder. I think I will. Can I zoom? Can you see this? Data files will have all of these. Do you have all the files? Okay.
Regarding this concept, how these things work in sex chromosomes? In the case of Y chromosome, I don't know, like I think it has less genes. So, if they selection one gene,
So sex chromosomes will have their own problems, but not the kind of problems that you're saying. The estimates of expected values will change in sex chromosomes because the effective size of the population that will have that chromosome is much lower than the total population size, right? If you are a diploid individual, the total number of autosomes that the whole population has is 4n, right? But the total number of sex chromosomes, X chromosome, not Y, X chromosome that this population will have will be 3n. And the total number of Y chromosome that this population have will be just 1n, right? And so the estimates of your expected value will change based on this effective population size. But no, so when effective population size decreases, the expected values increase because it's basically simulating a bottleneck, right? So observed is what you see. That is not in your hands. Only the expected is in your hands. We can discuss this further. Like there is one session free after this. Okay. So yes, can you upload the pi and the Tajimas-D file from the data? Also the frequency file. Now you'll see if you've uploaded this already, you will see that the pi and the Tajimas-D file has a lot of NaNs. That's an artifact of the tool. So wherever it finds an empty, so not all 26 individuals have been sequenced properly, right? There will be some individuals where neither the reference allele exists nor the derived allele exists. It's just not sequenced. And so when it tries to calculate Tajimas-D in those positions, it would not give out a value, it would give you an NaN. Right? So you've got to remove that NaN. And so the next command is to omit all the rows that have NaN in them. So you have to do this with both pi and Tajimas-D files. Yes. So the first sequencing run that we did was not that great. So I have to do this. Yes. So the filtration step that I showed right before this, the VCF tools, will have pass or fail for certain SNPs, right? The SNPs that fail will not be used for further analysis. And so this will be like an empty spot. Let's say you have 26 individuals, 5 of them have this empty spot and the rest have this whatever, reference or derived allele. When it tries to calculate Tajimas-D, it would say, I can't do that because not enough individuals exist. And so it would give you an NaN. So you have to remove that. That's an artifact of the tool. The next step is to count the number of variants that you have, total number of variants that you have. So in that file, in that Tajimas-D file, you have chromosome number, position, number of variants in that window, and the Tajimas-D value for that window. So if you sum up all of those n variants, you will find the total number of variants that are there in this region of VCF that I've given you. Mind you, I have not given you the whole genome. I have given you a very small region of X chromosome, because yesterday when Shweta was trying to demonstrate with just 80,000 SNPs, most people's machines stopped working. So I created a very small data set. Most machines will not work for the whole genome. So you need like a proper setup. I work on the cluster. So you can basically subset your VCF file into, let's say, 10 or 15 different VCF files and work separately. So how many variants do you have? Are you able to make that histogram? Yes, so what do you see? So what do you see? Do you see anything weird? Anything different from what Shweta was showing you yesterday? Yes. Why don't you have that here? What do you think is happening? This is a dummy data. I have created this specifically to teach you something like important. You will not usually see this in your data, but sometimes if there are problems, if there are specific regions that behave weirdly, you should be able to identify what this problem is. This step is called sanity check. You see, you are working with such large data sets that you can't actually see the data. You have to write a code to be able to see all of this. And so you have to predict what kind of problems might be there, and then write a code and find out if that problem exists. And so what exactly is the problem, and can somebody tell me why you see this? I hope you got at least these three. No? Can you raise hands if you are having trouble so that I can come and help you? Saying cannot open file. Oh, so can you just open the folder?
Yeah, this one.
So you don't have the data files. Can you just go back to R? Change the name from data files. Start with LG5. Just remove that bit. Yeah. Yes. Now try. You see, because the folder does not exist, right? You did not download the whole folder. You only downloaded the data files folder, and so it's not able to read that.
That's only what comes up in the data files. But
How
then
is
when
that?
I downloaded the data files alone, that's in my data files.
Oh,
So I set the working directory to the data files file.
okay. So then now change
Yeah.
this bit.
Yeah, so I changed that to this name,
Yes.
but it's still on.
Yes, because there is this data files directory that you have to go to, and it
Yeah.
can't find the data files. So just remove this bit. Right? There's no directory called data files because you're already in data files.
Oh,
Right?
okay, right, sorry.
So just try now.
Easy.
Try now.
Thank you.
Got it? Can somebody tell me why this is happening? So when I was working with these genomes for the first time, I saw this and I did not know what was happening, and I did not know this was even a problem. So I just went ahead and I told Deepa that this is the result. And then 10 days later, I was talking to a friend and I realized that this is the problem. Can somebody tell me what the problem is? To some extent, yes, but exactly what about samples? It is a sample size issue, yes. But you have 26 individuals, right? Something is happening within those 26 individuals that is giving you this signal. Can you tell me what that is? So if you go to your VCF file, you can't do that right now, but try doing it at home. you will see that this giant peak that you see at 0.5 is because of a certain region which is only there in 3 of your individuals. And so this giant peak is coming directly from those 3 individuals that are all heterozygous for that region. And so I've specifically given you a region of that X chromosome that shows this. And this is a problem, right? If you see this, that means you are selectively enriching a certain area for just some individuals and not the others. And so when you actually calculate Tajima's D there, you will see an enrichment of positive sites just because of this problem, right? And so you have to make this for all your regions to make sure that something like this does not happen. One way to correct for this is to have a proper filter in the beginning. You say anything that has missing sites or anything that is only represented by some of the individuals, you remove those sites entirely. And that will solve this problem. But I did not have that filter, right? I showed you the VCF tools commands. I did not have that filter. I just had a depth filter. And so this kind of a problems can still exist. Sorry, what? From 26 individuals, yes. So I have 26 individuals that are sequenced so that VCF files will have, so this VCF file will have 26 such columns, 0 or 1. So this is an individual. 0 means that there is a reference allele, 1 means that there is a derived allele, and you will have 26 such columns in that VCF file. So that's actually a weird question because it's actually a good question. The problem is that when you work with non-model systems, it's recommended that you use all the individuals together to call variants because more the number of individuals, the better the variant calling would be. But if you're working with something like humans or Drosophila or mice where you have a very good reference and you can get very good sequencing done, you can call variants individually. You don't have to call all 26 individuals together. So I work with a non-model system, so I used all 26 together. That is also something that I learned by hit and trial. That is also something that I learned by hit and trial. I did not know that this is a problem. So when you call individually, you will see a lot of missing sites because that single individual does not have enough depth of sequencing to be able to call that variant. But when you pool all those 26 individuals together, you have enough data to reliably say whether the variant exists or not. You can remove the individuals entirely. Yes, yes, yes. You can remove those 3 individuals and then work with all the other regions. Or you can remove that particular region and keep all the individuals. That's your choice. I have removed only those 3 individuals for the further analysis, but you can just remove that region and go ahead. Right? Are people able to plot these? And can people tell me what exactly is happening here? So this is pi. This is nucleotide diversity, the observed diversity, and you see this nice region where there's a very cool drop. Is that a signal of sweep? Did people reach here? At least just tell me that. Is that a signal of sweep? It's just one signal of sweep, right? But exactly. So just one signal of sweep is not enough because we discussed lots of these confounding factors, right? So it's always recommended that if you're looking for signatures of selection, you look at multiple signatures of selection and you only zoom in on regions that show positive signatures for all the things that you have measured. Because a single region can have confounding factors. This could be happening because of a bottleneck. You don't know. So if you look at LD in this exact region, and you look at site frequency spectrum in this region, and you see positive signals, you know that this is a true sweep. So it's always recommended that you use multiple signatures of selection when you're looking for actual regions under selection in the genome. There is a tool called Raised that can do this for you automatically. So it will calculate all of these 3 signals together. I think Krishnameg must have talked about this fleetingly. It's a very hard tool to set up, so I did not use that. I'm just using Tajima's D. VCF tools is very easy to install and work with. But somehow you could not get VCF tools also to work. Right on time, actually. I also wanted to discuss this, but it's really not that necessary. It's just, again, I have zoomed in on that. This is for a large region of the chromosome, right? I have zoomed in on this one region, and we are just looking at that to see whether the signal that we see is actually true signal or not. And you should do that. Like I told you, there can be problems that you can't even anticipate. And so it's always better to just go deeper and deeper and find out if whatever you see is true or not. Especially with data sets like this where you can't see anything. It's just a random ones and zeros. You don't know what is happening. Cool. Any questions? I have 2 more slides, but I don't have time. So if people are interested in divergence leading to selection, divergence and selection happening together, I can talk about this in the free time. And if people have experimental evolution systems where you want to identify signatures of selection in an experimentally evolved line, there is a way for that, and we can discuss this in free time. Cool? That's it. Thank you.